Assalamualaikum everyone, welcome back to this channel. So my name is Alia Wahid, and in this video I am going to tell the whole chapter of a virus in this video. So this is the microbiology, and this is the chapter number 3 from our syllabus. So now let's start the topic of virus. So first of all, I have defined the virus to you that it is obligate intracellular parasite. What is mean by intracellular parasite? So it is a parasite, mean it is preying on its host, and mostly its hosts can be animal, plant, and various kind of bacteria. So it is penetrating into their cells and infect them and cause a lot of diseases to them. So this is the definition, and it also contain a genome, which is could be either a RNA or DNA, cannot be both. Okay, so if I if a virus is present, so it contain whether a DNA or whether RNA. You will never observe a virus which contain both of RNA or DNA. Okay, so now what is the viron? It is written that it is a complete virus particle. So a complete virus particle which is fully matured and fully grown, this particle is called as viron, and its function is to transport your RNA DNA into your होस्ट सेल. सो अगर होस्ट सेल के अंदर अगर आपने इस वायरस का, जैसे ये डीएनए है, ये पेनिट्रेट करवाना है, तो आप उसको वायरॉन कहेंगे. वायरॉन का फंक्शन ही यही है. फिर प्रॉपर्टीज़ है दैट दे आर ऑब्लिगेट इंटरसेलुलर पैरासाइट. देयर साइज इज फ्रॉम 20 nanometer to 450 nanometer, and in their nature they are non-living things, and they depend upon the host. So they are outside a living body or outside the host, they are non-living, but when they go inside a living body, they will become active, and they will use the machinery of that host and produce their proteins. So they depend upon the host. They have no enzymes in them, they have no machinery. So therefore they use all the enzymes, all the machinery of that host, and produce their own components. Now let's see the what is the size of a virus. So size of a virus is range from 20 nanometer to 450 nanometer. So 20 nanometer is the smallest virus, which is the parvovirus, and for 50 nanometer is the largest virus, which is the pox virus, and its size is approximately equal to the size of smallest bacteria. Now let's discuss how to detect a bacteria or a virus. So there are some ways by which we can detect it. So the first is the electron microscope, second is the electron x-ray diffraction method, and last is the staining method. So staining method is preferable for the bacteria, but now it is also useful for detecting the virus. And there are two types of staining, positive staining and negative staining. So in the negative staining, you will deposit a opaque layer or thin layer of salt over the virus. So let's suppose this is a virus, you will deposit a thin layer of salt on it, and after depositing it, you can detect the shape of that virus. And with the help of positive staining, you can see the inner structure of this virus. Okay, so now let's discuss about the viral morphology. So your virus is mainly composed of two components, which is your core and your shell. So your core is consist of nucleic acid and genome, and the outer shell of that virus is called as capsid. Now we will discuss this virus morphology, and we will discuss all of these components one by one. So the first component is the genome and the nucleic acid. So a genome is your full genetic material of that particular virus is called as genome. So it could be either a RNA or DNA, but not both. So this is very important concept, and you have to fix this concept into your minds. Second thing, that you on the base of your nucleic acids, there are some types of viruses. Some viruses contain single stranded DNA, some contain double stranded, some contain single stranded RNA, and some contain double stranded RNA. So we will discuss them, all of them, and also we have discussed some examples of them, so in order to identify to you. Now let's move on to the structures or the types of viruses on the base of their genome. So here is the types of viruses on the base of their genome. So there are two types of viruses: isocytral virus and helical virus. So what are them, and we will discuss them when we discuss the capsomere. Okay, so therefore the your isosaderal virus is due to the shape of that genome. So it is in the form of 3D structure, and in the helical virus your genome is in the form of helical, is in the form of coiled. Okay, so the coiled form is just like this, as it is here, given here. It is just like this, so nucleus is going to be coiled. But as you can see, this is the isostrahedral genome, which is in the form of coiled or condensed form. Okay, now let's discuss the second major part of your viruses, which is the capsid. Capsid is the protein shell which is enclosed outside your virus genome. So this capsid is made up of a lot of subunits, and these subunits are called capsomere. And their function, what is the function of this capsid? So it is involving in the protection of that particular virus, it is involving in the attachment of that virus to the host membrane, and lastly it is used for the penetration of your genome into your host cell. Now let's explain all the things about the capsomere. So what is the capsomere? So there are some this is the protein subunit by which your capsid is made. Now there are two types of capsomere: helical capsomere and isocytral capsomere. So your helical capsomere is in the form of rod shape. So as you can see here, this is the helical genome. Now this helical genome is enclosed by here. So various kind of circles of subunits of capsomere surround itself in the form of rod shape. Okay, and then they will enclose by a shell, and this shell is called as this envelope. So this is the envelope of envelope. Okay, now isocedral isomer is this, which is just similar to a benzene ring. It is a 3D structure, polygon shape, having 20 sides, 12 corners, and 20 faces. Okay, so it has a 3D structure. So there is difference between the arrangement or the symmetry of your capsomeres. So this is the isocedral capsomere having envelope in them, and they have various kind of spikes on them. These spikes are made up of glycoproteins. Now let's discuss what is the nucleocapsid. So your nucleus capsid can also be called as genocapsid. Okay, so they are different. So nucleo word mean nucleic acid, and capsid mean capsid. So your nucleic acid, this is your nucleic acid, and this is your capsid. So nucleic acid plus capsid is forming, which is called as nucleocapsid, and can also be called as genocapsid. Geno mean genome, and capsid mean capsid. So it also includes this. Now the last morphology of your virus is the envelope. So envelope is the surrounding around the capsid. Okay, so some viruses has this envelope, but some has no envelope on them. So this envelope is actually a flexible membrane around them. It is composed of your lipid, your protein, and some it is consist of glycoproteins. The spike of them are composed of glycoproteins. They are just similar to the lipid bilayer of your host membrane. And there are two types of viruses on the base of the presence of envelope. So some are enveloped viruses having the envelope over them, and some are non-enveloped, can also be called as naked virus. So they have no envelope in them. And what is the function of this envelope? It's very simple, that it is going to protect. Function is that it is going to protect your virus, and second thing, it will never allow your host cell to detect that it is a virus. So it is decreasing its chances of recognizing by the host cell. Okay, so now we are going to move toward the classification of your virus. So here the classification of virus is given. So your virus is classified on the base of their genome and on the base of their site of disease. So first of all, let's discuss their types on the on the base of their genome. So whether they contain DNA virus or whether they contain RNA virus. So DNA virus is containing DNA as a genome, and RNA virus is containing RNA as a genome. Now let's discuss the classification of DNA virus. So DNA virus can be enveloped or can be non-enveloped. Now, the enveloped virus only contain double stranded DNA. Okay, they never contain a single stranded DNA. Okay, so now what is the example of them? So, herp virus and pox virus are the double stranded DNA enveloped DNA virus. So, now in your syllabus it is explained that you have to classify the virus and have to explain at least one member of this classification. So I have explaining all of these members one by one. So now in the pox virus, this is from a family of a virus which is called as pox viridae. So always remember, whenever the word end with viridae, it's mean this is a family name of that virus. So they are causing the smallpox disease to you, and in order to treat a smallpox you will use a vaccine which is called as vaccina. So it is given to the pox patients in order to treat them. Now let's discuss the non-enveloped DNA viruses. So they can be of two types: double-stranded DNA and single-stranded DNA. So the double-stranded DNA example is the adenovirus. So adenovirus is from a family which is the adenoviridae. Okay, and they are causing you a respiratory infections in the human beings. Last is the single stranded DNA. Its example is parvovirus, and parvoviridae is its family, and it is causing gastroenteritis. Gastroenteritis means inflammation into your GI tract. So they are responsible for this disease inside the human body. Now, after this, we are going to discuss the classification on the base of site of disease. So RNA virus will be discussed here. So just wait for a second. Now let's discuss the on the base of site of disease. What are the classification of virus? So this is actually on the base of their transmission and on the base at which they are causing the disease inside the human body, like pneumonia tropic. So this is the group of viruses, and these are the diseases which is caused by them. So pneumonotropic, as the name indicates, they are causing infection inside the respiratory tract. Examples of them are influenza and adenoviruses. Okay. Next is dermotropic. So derma means skin, tropic mean causing infections on them. So they are acting and causing disease on the skin. They are smallpox and herpes simplex and measles, mumps, and lot of examples of them are available. Next is the viscerotropic. Viscero mean internal organs or deep organs of your body. So they are causing infections here like yellow fever, dengue fever. So they are causing infection inside or deep into your body. Next is the neurotropic. So they are causing some kind of neurological disease to you. So there are herpes virus. This herpes virus is released from an animal, and there is the polio virus. So these are the examples of them. Now let's discuss the last classification of virus, which is the RNA virus, and see on the next slide. So here the classification of RNA virus is given. So again, the RNA virus is divided into two parts. First is the enveloped, and second is the non-enveloped. So here envelope is consists of further two divisions: single stranded RNA and a single stranded RNA with reverse transcriptase. So reverse transcriptase is actually an enzyme which is present inside the virus. So here, as you can see here, this is the genome, and the dot which is written here, this dot. This dot represents the reverse transcriptase enzyme of that virus, and so they are present in them. And the single-stranded RNA viruses which contain this reverse transcriptase, they have given a separate family name of them, which is this retrovirus. So retroviridae is the name of their family, and they are causing their example includes the HIV. So you know very well about HIV virus, immunodeficiency virus. So it is causing the AIDS disease to you. Now let's discuss the single stranded RNA. So it composed of CV and NSV. So CV means segmented virus and non-segmented virus. So there are some kind of viruses who have segmented RNA. Segmented mean the RNA which is divided or cut it into the parts. So now the examples of them is the orthomyox virus, and the non example of non segmented is RHABDO virus, ठीक है. So Rahabdo virus. Now let's discuss about the RNA viruses of non-enveloped. So they could be double stranded or they could be single stranded. So single stranded are on the both side. Just the double stranded is in the non-enveloped. While in the case of DNA virus, the double stranded is in the case of both enveloped or non-enveloped. Now the examples of them is the Rio virus. Rio viridae is the family and they are causing the respiratory infections to you. Last the single stranded RNA is the Pyrona virus and they are causing Pyrona viridae is the family of them. Now let's discuss and classify the virus on the base or at the site or at what kind of organism they are causing the diseases. Like animal viruses are causing diseases in animals or humans. Plant viruses causing diseases in plants. Bacteria viruses causing diseases in the bacteria, so they can also be called as bacteriophages. So example of animal virus is polio, herpes virus, rio virus, dengue virus, something like that. The plant viruses examples is not given. Bacteriophages examples is from T1 to T6. So T1, T2, T3, T4, T5, T7, and 6 and T7. So they are the bacteriophages which are infecting mostly E. coli inside your body. Now lastly we will discuss about the replication of your viruses. So the first process is the attachment. So what happens, this is the bacteriophage which is going to attach at the bacterial cell. So this is the bacterial cell wall and this is the bacteria chromosome. Now the first step is the attachment. So it is going to be attached with it. There is occurring a weak union between them. A chemical attraction happens between them. So now the next step of your application is the penetration. So now what is going to be happen, this side of your bacteriophage will release an enzyme which is called as lysozyme. So it dissolve this cell wall. So now this cell wall is going to be dissolved like that. A whole formation occurs here, and after that your DNA, okay, which is present here, it passes throughout and will release such a thing like that. After the release of this DNA, now this DNA will go and incorporate into your nucleus of your chromosome. And after that the third step happens, which is the biosynthesis. Now it is going to be disturb the host chromosome. It is disturbing the messenger RNA and going to make its own enzyme, its own ribosome, and its own amino acids. And then it also form its capsomeres. So the biosynthesis for the viron components will happen. Then after that there is the maturation. So in the maturation, your the head and tail of your bacteriophage. So this is the head of bacteriophage, this is the tail. So inside your bacterial cell, this head will form or mature itself, the tail of it will mature itself and form like this, okay. So the components of virus is going to be happen in formed inside the cell. Last step is the release. So now at the release, your bacteria cell will burst itself. Now it is bursting and releasing the full formed viron in the outside of the environment. So now this is the full virus and it is released into your outside environment. Then, and one thing you have to remember that 50 to 200 new bacteriophages will form and eliminate outside. And what is the time, after what time, after 20 minutes this happens. So this is the lysis stage at which your bacteria cell burst and it will release all the things out of it. So this was the step for the replication of your virus. Next is the transmission of virus, that how the virus transmitted. So if you have a respiratory way, so it will be transferred through coughing, sneezing, like influenza virus is separate by it. Next transmission is through the fecal or oral route. So enterovirus is going to be transmitted by this way, and bloodborne. So they are transmitted through the transfusion of blood and something. So HB virus, hepatitis B virus is separated by it. Last is the animal vector. So some viruses are separate due to animals, such as rabies virus, which is separated by a dog which is infected with it, and when this dog bite a person, it will cause rabies fever to that person. Lastly, the treatment of virus is given, you will give antiviral therapy to that patient, antiviral drugs will be given, some kind of vaccines will be given to that patient in order to treat them. So this was all about the chapter of your viruses, and we have discussed all of them. We have covered all of the chapters of it and all the topics which are included in it. Last, the topic of virus is remaining, which is the cultivation of viruses. So the cultivation of virus will be discussed in the next lecture. I hope that you will like this video. If you like it, then please press the subscribe button, press the bell icon in order to get the notification of all the videos which I uploaded. So thank you so much for subscribing to my channel. Meet you soon in the next lecture. Till up, Allah Hafiz. Take care of yourself.